All right. Okay, to all the grade 12 life sciences learners who are watching today or who will be watching this presentation today, I just want to say good afternoon to you all. So today I'm going to do exam preparation for trial examination paper 1 that you will be writing on the next coming Friday, which will be on the 3rd of September 2021. So actually what I'm going to do is I will just focus on the key points. So it means that all those basic things that will make you or that will help you to pass the trial examination as well as the final examination. So let us move straight into our presentation. So now we are on the first slide. Welcome aboard. We appreciate you all for watching today. Okay, so like as I have said that our presentation will be based on the key points of paper 1. And then, but first, before we move to the key points, and then let us start again reminding us of how our paper 1 format looks like. So remember that we have section A and section B. And then section A, it is still out of 50 marks, and then section B, it is out of 100 marks. So it means that section B consists of 2 questions, question 2 and question 3, and then both they are out of 50 marks each. And then on the next slide, this is what we have, the distribution of topics for September and November 2021 examination. So let us look at the first topic, reproduction in vertebrates. So we know that it is, or it will be out of 8 marks. And then human reproduction will be out of 41 marks, responding to the environment, humans or human nervous system. It will be out of 54 marks. And then responding to the environment, which are plant hormones, and then it will be 13 marks, and then endocrine and homeostasis, and then both will be out of 34 marks. All right, and then we move to the next slide, and then this is what we have. Drill the following. This is basic, especially for level 1 to 3 learners. Like as I have said that today's presentation will be based on the key points that will help level 1 to 3 learners, especially level 1 learners, to pass the trial examination and also the final examination for this year. So the first topic that we have, guys, it is reproduction in vertebrates, and then we know that reproduction in vertebrates, it is out of 8 marks. So please just make sure that you revise the following: external fertilization and internal fertilization. And then bullet number 2, we have ovipary, ovovivipary, and vivipary, amniotic egg, precocial and altricial development, and then parental care. So guys, I know that you don't have a problem with bullet number 1, number 3, number 4, and number 5. But most of you, you are struggling with bullet number 2. So let us quickly look at a summary of bullet number 2. So please remember that ovipary, with ovipary, and then this is what we have. It is a method of reproduction in which eggs are laid and embryos develop outside the mother's body but within the egg. And then amniotic egg is a type of egg that is produced by oviparous birds and egg-laying mammals. So please remember that it is a method of reproduction in which eggs are laid and embryos develop outside the mother's body. So it means that the development of the embryos will be outside the body of the mother. Then the second one, let me skip the second one, and then we move to the third one, which is vivipary. It is a method of reproduction in which the fetus develops in the mother's uterus, and it is nourished through an umbilical cord. And I know that you don't have a problem with this one. And then let us go back to the second one, ovovivipary. And then this is the strange one. And then it is like this. It is a method of reproduction in which young develop from eggs that are kept within the mother's body, but separated from her body by the egg membranes, and then they are born live. Okay, and then on the next slide, we still have these reproductive strategies, and then ovipary, again, eggs develop and hatch outside the female's body. And then fertilization may be external or internal with this one. And then vivipary, embryo is attached directly to the female's uterus through the placenta and umbilical cord. Ovoviviparies, please remember that eggs develop and hatch inside the female's body. So please make sure that you go through or you revise those reproductive strategies, because they will be there on your next coming examination, on your trial examination, as well as your on your final examination. And then mark allocation for that, like as I have said, that it will be 8 marks. So on the slide, we have an example of one of the questions. And then this question, it is from one of the previous question papers. So let us look at this question. And then it is like this. Question 1.5. The diagrams below represent organisms with different reproductive strategies. So we have diagram number 1, number 2, and number 3. And then when we look at diagram number 1, we have the male fish and the female fish. And then below the male fish, we have the egg cells. And then female fish, those small dots. And then they represent the sperm cells. So let us try to go through these questions. Question 1.5.1. Which diagram or diagrams 1, 2, or 3 represent organisms, number A, where external fertilization takes place? And then if you go back to your diagrams 1, 2, 3, where external fertilization takes place, and then we will say that it is diagram number 1. Why? Look at the sperm cells and the egg cells. So it is clear from this diagram that fertilization will take place externally. So that is diagram number 1. And then number B, where extra embryonic membranes develop to assist with the protection and nutrition of the embryo. And then we go back to our diagrams. It will be diagram number 2 and number 3. Number 2 and number 3. And then I know that you still remember when we talk about the extra embryonic membranes, we are referring to membranes such as chorion and amnion. And then number C, which is or are oviparous, and then it is out of 2 marks, so it means that you have to mention 2 diagrams there. Then it will be diagram number 1 and diagram number 2. Remember what we said about oviparous? Let me go back. It is a method of reproduction in which eggs are laid and embryos develop outside the mother's body within the egg. And then an example of this, it is an amniotic egg. And then if you, even if you look at diagram number 1, eggs will be laid, and then diagram number 2, and then both the diagrams, they represent what? Oviparous. And then question 1.5.2, name the type of the egg produced by the organism represented in diagram number 2. And then it is out of 1 mark. And then the answer for this question will be the amniotic egg. So here are the answers of those questions. So please, guys, please just make sure that you revise those reproductive strategies so that you will be able to get that full 8 marks. They will be there on your trial examination, and then again you will also see them on your final examination. And then let us move to human reproduction, which is out of 41 marks. And then the key points here under human reproduction. So please make sure that you know or you revise the structure of the male reproductive system and together with the female reproductive system. So it means that you have to know all those labels which are stipulated on your examination guideline and the functions of those labels or the parts. Okay. So this is the structure of the female reproductive system. So, and then again, please make sure that you know all these parts and the functions of the parts. And then again, we have the structure of the male reproductive system. Make sure that you know all the parts which are stipulated on your examination guideline together with the functions of all those parts. And then another thing which is very important with this one, it is the glands. So please make sure that you know your glands, know where they are located. Look at the first gland, the seminal vesicle, and then look where it is located. And then also we have the corpus gland. And then again, we also have the prostate gland. So please, 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 this is where you will also collect the marks. Just know where your glands are located or situated, and then after that, you have to be able to mention the function of each and every gland. And then another thing which is important here, look at this part, the epididymis. Please know the function of the epididymis. And then another thing which is also important, the testes and the issue of the scrotum, please. Okay, on this slide, we have the functions of all the parts, okay, which appear on our diagram. And then all these parts, again, they are also there on your examination guideline. Like as I have said, please make sure that you know the location of all these parts together with their functions, not forgetting the glands, seminal vesicle, corpus gland, and prostate gland, please. And then again, the issue of the scrotum. When you look at this diagram, this is what we have. They say that the skin sac, a scrotum, it is a skin sac that protects the testes and it holds the testes outside the body at a temperature that is 2 degrees Celsius below 37 degrees Celsius, which is the temperature of the body temperature of humans. So this is the best temperature for the production of the sperm cells. All right. And then on this slide, this is what we have. We have both the diagrams or the structures of the male and the female reproductive system. And then after that, let us try to go through these questions. Okay. So question number 1 is like this, 2.2.1. Name the parts D and G respectively. Let us look at D. Here is D. And then the name of this part, it will be the ovary. And then G, here is G. And then the G, it represents the urethra. Please note ureter. That is the urethra. And then question 2.2.2, give one function of each of the following parts. Number A, part number A. Here is part number A. And then I'm sure that you still remember that part number A, it represents the fallopian tubes. So what is the function of the fallopian tubes? Fallopian tubes transport the immature ovum as well as the developing zygote in the direction of the uterus. The function of the fallopian tube, it is to transport the immature ovum as well as the developing zygote in the direction of the uterus. So that is label number A, which is the fallopian tube. And then let us look at number B. Structure number B. Okay, here is number B. So number B, the function of number B, this is where the embryo or fetus develops from implantation to birth. So that is the function of number B. So the next question, question 2.2.3, give the letter that represents the endometrium and name the 2 changes in the endometrium. And then let us go back to our diagrams. The letter that represents the endometrium from our diagrams, it will be letter number C. And then our question continues like this. Name the two changes in the endometrium caused by hormones. So which are these hormones? It is estrogen and progesterone. So we want the two changes in the endometrium which are caused by estrogen and progesterone to ensure the implantation of the blastula or blastocyst. So let me check or show you how to answer this question. So like as we have said that the letter that represents the endometrium from our diagram is letter number C. Look at question 2.2.3. So the two changes in the endometrium which are caused by the hormones, and then hormones which are progesterone and estrogen, and then to ensure the implantation of the blastocyst. And then this is how you have to answer the question. It will be, you will say that the endometrium, it becomes enriched with glands and blood vessels, or it is thickened, or it is thickened with gland and blood vessels, or richly supplied with glands and blood vessels. Okay, then we move to the next question, question 2.2.4. Explain the significance for human reproduction of the position of part I. Here is part I in relation to the rest of the body. Question 2.2.4. So this is how you are supposed to answer this question. The scrotum carries the testis outside the body, where it is 2 to 3 degrees Celsius below the body temperature to ensure optimal sperm production. So question 2.2.5. Question 2.2.5, give the letter and the name of the part in males which is responsible for each of the following functions. And then let us look at the male reproductive system. Okay, and then number A says stores sperms. So that will be letter number H, and then the name of H is epididymis. And then the letter in males which is responsible for each of the following, then we move to number B, is fluid protects the sperm against low pH levels. So number B will be F. Let us look at F, and then F, it represents prostate gland. Like as I have said that, so please make sure that you know your glands, know those glands and the functions for each. All right. And then we move to the next slide. We have the structure of the sperm cell. And then you know that from your examination guideline, it is clearly stated that you have to know the structure of the sperm cell. You have to know that structure and be able to label it and give functions of the parts. So this is the structure of the sperm cell. And then we have the head here. And then this part, it represents the acrosome. And then we have the mitochondrion here on the middle piece. And then we also have the tail. All right. And then again, we are also expected to know the diagram or the structure of the ovum, and together with all those parts which are mentioned or stipulated on your examination guideline and the functions of those parts. Okay, and then we have a question which is question 1.4, and then it is based on the structure of the ovum and the sperm cell. So we have label number A, B, C, D, E, and F. Question 1.4.1, it says identify part number A. What is part number A? And then here is part number A, which is from the ovum, and then part number A, it is a jelly layer. So part number B, Here is part number B, and then it represents the cytoplasm or cytosol. And then part number C, it is the acrosome from the sperm cell. And then question 1.4.2, name the process involving meiosis that leads to the formation of an ovum. And then the name of that process, it is oogenesis. 1.4.3, write down only the letter of the part of the sperm that enters the ovum. So that will be part number D, which is the nucleus. Write down, question 1.4.4, write down only the letters of two parts that enable the sperm to move towards the ovum. So we have part number E. So please remember that part number E, it is the mitochondria, and then it contains which supply the sperm cell with energy. So that will be part number E, and also part number F, which is the tail. All right, and then we continue. So on our next slide, we have the process of gametogenesis, and then which is divided into spermatogenesis and oogenesis. So please, learners, make sure that you know these two processes before you go to your trial examination, and then again also to your final examination. And then another thing which is very important, please know that, or just remember that you are not allowed to describe or to discuss these two processes using your own words. So it means that starting from today, if you are not sure about these two processes, just make sure that you drill and you drill and you drill them. They are always there on your examination. So oogenesis, spermatogenesis, and then hormonal control of the menstrual cycle, which consists of, it is based on the ovarian cycle and uterine cycle, and then with reference to the action of FSH, estrogen, LH, and progesterone. So please just make sure that you know your four hormones, which are FSH, estrogen, LH, and progesterone. And then once you know these four hormones, and then automatically or already you will be having 15 marks in your pocket. And then another thing which is very important, learners, please remember the negative feedback mechanism involving FSH and progesterone. Please, this thing, the negative feedback mechanism, it is always there on your examination. It will be there on your trial examination, and then again you will also see it on your final examination. So please just make sure that you know your hormones, your four hormones, and then after that, negative feedback mechanism. You have to drill this process. Okay, we have a diagram here which is based on the hormonal control of the menstrual cycle. And then let us look at this first row. We have the ovarian cycle here. We have a primary follicle, and then it grows and grows, or it becomes bigger and bigger under the influence of FSH. And then up until it reaches this stage where we are now having the Kraffian follicle. And then remember that the Kraffian follicle, it will burst here on day 14. And then it will release an ovum, and then by the process of ovulation. So this is what you see here. And then after that, under the influence of the hormone LH, that Kraffian follicle, which has ruptured, you still remember that now it will now turn into corpus luteum. And then if you look at this diagram, look at the size of this corpus luteum, and then the next one, now it becomes smaller. So it means that it is now degenerating. Why? It simply means that fertilization did not take place. So menstrual cycle, like as I have said, that it is divided into two. And then it is the ovarian cycle and uterine cycle. And then again, look at this diagram of the ovary. All right, we have the primary follicles here, and then they will grow under the influence of FSH, or follicle stimulating hormone. And then now an ovum will be formed inside the Kraffian follicle. And then remember again, Kraffian follicle, it also produces or secrete estrogen. and then here ovulation takes place, and then after that we are having the corpus luteum, which will secrete progesterone, and then the last diagram, this one, it is still the corpus luteum, but now it shrinks. And then below here we have the uterine cycle, then which represents the lining of the endometrium. Okay, events of the ovarian cycle, the same thing here. And then this diagram, it is from one of the previous question papers. And then again, primary follicles, and then Graafian follicle with the ovum inside, and then it bursts, and then the ovum will be released by the process of ovulation, and then after that we have the corpus luteum, and then which secretes or which will secrete progesterone, and then look at the second one and then the third one, and then it means that it is now degenerating. Okay, the lining of the endometrium again. And then again we have our 4 hormones, FSH on this side, and then LH here, and then here is estrogen, and then in the uterine cycle, and then also we have progesterone. Okay, like as I have said that please just make sure that you know the negative feedback mechanism between FSH and progesterone. You will see this thing on your next coming examination, on your trial examination, as well as on your final examination. And then let us take this question. It is question 2.1 from one of the previous question papers. And then it reads like this. The diagram below represents the sequence of events that takes place during the ovarian cycle of a female. So here is our diagram. We have label number A, and then label number B, and label number C. 2.1.1, give the name of the number A, hormone that controls the development of structure number A. And then the name of that hormone is the follicle stimulating hormone, which is FSH. And then process taking place at C, this is ovulation, the release of the ovum after the Graafian follicle has ruptured. 2.1.2, describe the change that takes place in the uterus as the result of the hormone secreted by structure A. Describe the change that takes place in the uterus as the result of the hormone secreted by structure A. And then what is the name of that hormone? That is estrogen. So it will cause the endometrium to become thicker or more glandular or more vascular. Or it will become thicker with glands and blood vessels. And then question 2.1.3, if structure B degenerates, so remember that structure B is corpus luteum. It degenerates if fertilization does not take place, and then explain the implications of this for the ovarian cycle. So it simply means that you have to explain what will happen if corpus luteum degenerates. Number A, inside the ovary, and then number B, inside the uterus. So this is how you have to answer this question. I'm just going to show you. Okay, that is question 2.1.3. And then let me read the question again for you. Structure B, and then like as I have said that structure B, it is corpus luteum. It degenerates if fertilization does not take place. And then number A, explain the implications of this for the ovarian cycle. So it simply means that in number A, if structure number B, which is corpus luteum, degenerates, and then the level of progesterone will drop. And then after that, you have to ask yourself, and then what will happen if the level of progesterone drops? So therefore, FSH secretion is no longer inhibited or will no longer be inhibited, or FSH secretion is resumed, or FSH secretion will start. And then after that, if FSH secretion is no longer inhibited, and then what will happen after that? So it means that new follicles will start to develop. So that is what is going to happen inside the ovaries if corpus luteum degenerates. And then number B, again, what will happen? You have to explain the implications. If structure number B, which is corpus luteum, degenerates, what will happen inside the uterus? So again, the level of the progesterone will drop. And then if the level of progesterone drops inside the uterus, then what will happen? Therefore, the endometrium will no longer be maintained. And then that will lead to menstruation, or menstruation will take place. So please, that is how you have to answer that question. And then let us move to the next slide. And then on the next slide, this is what we have, the development of the zygote up until it reaches the stage of implantation. So please, again, this is very important. This is very important. If you don't see this thing on your trial examination, and then definitely you will see it on your final examination. But please just make sure that you don't go to the examination on Friday if you have doubts about on this. So, and then let us look at this. I'm going to show you how to answer this, or how to write this process during the examination. And then another thing which is important here, you also have to know the functions of the following: chorion, chorionic villi, amnion, amniotic cavity, amniotic fluid, and then umbilical cord and the placenta. And then if you look at this diagram, again we have the placenta. Please make sure that you know the role of placenta. The role of placenta: umbilical artery, umbilical vein, amnion, and amniotic fluid during gestation. Okay? And then this is what we have on this slide: development of a zygote until implantation. And then when you look at this diagram, we have the sperm cell here and the ovum, or the egg cell, and then you know that fertilization will take place, and then after that we will have the zygote. And then zygote will divide by the process of mitosis up until it reaches this stage called morula. And then normally we call morula a ball of cells. Why do we call it a ball of cells? Because even if you look at it, it is round, it looks like a ball, and then inside we have those cells. So that is why we say it is a ball of cells called morula. And then morula will also divide by the process of mitosis to form the blastula, which is a hollow ball of cells. So why do we say hollow with the blastula? It is because of this hollow space which is found inside. The same diagram again, fertilization here, and then after that we have the zygote, and then mitosis will take place, morula, the blastula, and then after that the outer layer of the blastula. Please remember that it will form a membrane which is called the chorion, and then the chorion will form the chorionic villi, and then after that implantation will take place. So again, learners, please just make sure that you know this process. So if you look on this slide, I have indicated here that how are you expected, or how do we write this in exams? This is very important. And then again, please make sure that you drill and drill and drill this process. From the diagram, again, I will repeat again, the sperm cell and the ovum, fertilization taking place, and then we have the zygote, and then zygote dividing by the process of mitosis to form this morula, ball of cells, and then mitosis again, and then blastula, which is a hollow ball of cells, and then after that, we have implantation here. You still remember that the outer layer of the blastula will form a membrane which is called the chorion, and then the chorion will form the chorionic villi, and then after that we will have implantation. So please, this is how you have to write this. Zygote undergoes mitosis Or you can also say that the zygote divides by the process of mitosis, and then after that to form a ball of cells. So normally for writing this, you will get 1 mark here, ball of cells, called morula. So please remember that once you say that, let us say you make a mistake, and then you say that the zygote divides by the process of mitosis, and then after that to form a hollow ball of cells called morula. And then it means that you will lose 2 marks, because morula, it is not a hollow ball of cells, but it is a ball of cells. And then after that, morula will also divide by the process of mitosis to form a hollow ball of cells called blastula. And then again, once you make a mistake and called the blastula a ball of cells, then it means that you will also lose 2 marks again. So please make sure that when you write this process, and then there should be logic. And then after that, the blastula will form the outer layer called chorion, and then chorion, which will form the chorionic villi, and then after that, implantation will take place. So please make sure that you drill and drill this process, because it is always there on your examinations. Okay, and then like as I have said that, again, you also have to know the functions of the chorion, chorionic villi, amnion, amniotic fluid, and then the placenta. So please make sure that you revise these processes. Don't go to the examination on Friday if you don't know the functions of these parts. And then umbilical cord, again, you still remember that it has the vein and the arteries. And then again, you have to know the functions. And then other functions of the placenta. So meaning that the role of placenta during gestation. So please make sure that you know all these things. Role of placenta during gestation, and then also the role of amnion or amniotic fluid and the chorion, please. So we have a diagram here from one of the previous question papers, and then the diagram below represents a sequence of events that may take place inside the human female reproductive system. And then, okay, question number 1.4.1 is like this. Identify the process taking place at 1 in the diagram above. And then let us look at 1. So we have the sperm cell and the ovum or the egg cell, and then the name of the process, it will be fertilization. And then 1.4.2 state the type of cell division. You remember that we have 2 types of cell divisions, and then which are meiosis and mitosis. So the type of cell division that takes place at diagram number 2 in the diagram below. So remember that we have said that the zygote will divide by the process of mitosis to form a ball of cells called morula. So it simply means that the name of the cell division here, it will be mitosis. 1.4.3, name 2 functional extra-embryonic membranes that are produced by structure C. So structure C, which is a hollow ball of cells, and then which is called the blastula. You remember that I have said that the outer layer of the blastula, it will form a membrane called a chorion. So this is the chorion, those 2 functional extra-embryonic membranes, and then they are chorion and amnion. So still on that question 1.4.4, it is like this. Identify the stage of development indicated by A. Here is A, and then this is the zygote, and then B, morula, and then C, blastula. 1.4.5, name the part of the female reproductive system where the events in the diagram above usually take place. And then I'm sure that you still remember that all these events are taking place inside the fallopian tubes. And then 1.4.6, give the chromosome number of the cell at A. Here is cell number A. If this cell, so you still remember that cell A should have 46 chromosomes. But the question is like this: if this cell is going to develop into a child with Down syndrome. So it means that the chromosome number now, it will be 47. So there will be an extra chromosome. So that is why we will have 47. And then let us move, okay, to the next question, question 2.4. Then 2.4. And then let us look at that question. Okay, the diagram below represents a developing fetus in a human body. 2.4.1, and then it says identify number A and then also number C. So let us look at number A, the outer membrane, it is the chorion, or also if you say the amnion, you'll still get marks. And then number C, where is C? And then C represents the umbilical cord. And then 2.4.2 state two functions of the fluid in part number B. So here is part number B. Okay, the name of the fluid inside here, I'm sure you still remember that it is the amniotic fluid. And then let me show you how to answer this question. So the function of the amniotic fluid, and then I like this. It protects the fetus from shock, protects the fetus from drying out, and then it also protects the fetus from temperature changes. And then the last one, it allows free movement of the fetus. So these are the functions of the amniotic fluid. All right. And then question 2.4.3. Name one system in the baby's body that takes over the function of part number D. Here is part number D. Once the baby is born. So this is how you have to answer this question. Okay, let me repeat the question again. Name one system in the baby's body that takes over the function of part number D once the baby is born. And then those systems are gaseous exchange system, and then excretory system and digestive system. All right. And then question 2.4.4, explain one negative impact on fetal development if part number D, here is part number D, is reduced significantly. All right. So if part number D is reduced significantly, so it means that the fetus will receive less nutrients. And then what will happen if the fetus receives less nutrients? And then therefore it will have a lower birth mass, or physically underdevelopment, or physically underdeveloped, and mental underdeveloped, or mental underdevelopment. And then again the fetus will receive less oxygen again, and therefore have a lower birth mass. And then waste will accumulate. And then what will happen if the waste accumulates? And then it will affect the functioning of the fetus. So that was question 2.4.4. And then while we are still, you still remember that I have said that our presentation today, it is based on the key points. The key points, the basic things that will help, especially the level 1 to 3 learners, to pass the trial examination. So on the next slide, this is what we have: response to the environment, and then humans, which is human nervous system. So please remember that you have to know the location and the functions of the following parts. And then under the brain, cerebrum, corpus callosum, and medulla oblongata. So please make sure that you know the location and function of those parts. And then again, we have the spinal cord. And then again, the structure and functions of the parts of the sensory and motor neurons, they are very important. So make sure that you know both these structures. And then again, the parts. You have to be able to identify the parts, which are nucleus, cell body, cytoplasm, myelin sheath, axon, and dendrites. Okay, we have the neuron here. So this is a simple diagram of the brain, like as I have said that, so please just make sure that you know the functions of all these parts, especially the cerebrum, corpus callosum, and medulla oblongata. You will go through that one. And then question 1.6, we have question 1.6 here, which is based on the structure of the brain. The brain acts as the main coordinated center for nervous activity, and then it receives information, interprets it, and responds accordingly. So we have label number A, B, C, and D on this diagram. And then let us look at the questions. Questions 1.6.1, and then it is as follows. Give the letter only of the part of the brain that controls the following. Number A, breathing. And then that will be part number C. And then B, memory. That will be part number A. 1.6.2, the part labeled A, here is part number A, is made up of two similar halves. How are these two halves attached to each other? So you still remember that the two halves, they are attached to each other by what is called the corpus callosum. So please remember, this is the corpus callosum on the brain, not the corpus luteum. So that will be the corpus callosum. And then 1.6.3, state one way in which the brain is protected. So please remember that the brain is protected from injury by, number one, the skull, meninges, and cerebrospinal fluid. And then let me show you the answers. Okay, the skull, the meninges, and cerebrospinal fluid. And then question 1.6.4, state two reasons why humans need a nervous system. And then let me show you how to answer this question. The nervous system is needed to detect changes in the environment, external and internal, and to react to these changes, as well as coordinating the various activities of the body. So please remember that you are also expected to know this. All right. And then we move to the structure of the neurons. Like as I have said that, please remember that you have to know both these diagrams, motor neuron and sensory neuron, as well as all these parts and the functions of these parts. Okay, and then we have questions here, which are based on these two types of the neurons. And then question 1.4, it is like this. The diagram below represents the structure of a neuron. Okay, we have this diagram and then label number A, B, C, D, E, and F. Question 1.4.1 says, name the type of neuron in the diagram above. Remember that I have said that you have to revise both the motor and sensory neuron. So you will have to be able to identify whether this diagram, it represents the motor neuron or the sensory neuron. So the answer for 1.4.1, it will be the motor neuron. 1.4.2, identify part number B, F, and A. So part number B, let us look at B. B, it represents the nucleus or nuclear membrane. And then F, here is F. F, it is this space, and then it is the cytoplasm. And then A, dendrite. Question 1.4.3, give the letter and the name of the part that number A transmits impulses away from the cell body. Question 1.4.3a. That is C. And then what is the name of this part? It is the axon. And then the second one, insulates and speeds up the transmission of the impulse. And then when you look at our diagram, it will be part number D. And then remember that part number 3 represents myelin sheath. And then 1.4.4, name the condition caused by the progressive degradation of part number D. so meaning that myelin sheath. And then the name of that condition, you still remember that it is multiple sclerosis. Okay, and then let us move to another concept. Now we are having the functioning of a simple reflex action, and then by using an example. So please, learners, you have to make sure that you know this diagram of a reflex action, and then again you have to be also to explain this, the movement of the impulse from the receptor up until it reaches the effector, by using any other example that you can think of. And then again, another thing which is very important, you have to be able to identify the parts on this. Remember that we have the receptor here, and then the sensory neuron, and then interneuron, and then motor nerve fibers and effector. Maybe you will get a diagram, and then after that you will be asked to identify some of the parts here. So you have to be able to do that. Okay, another thing which is very important, please, or which is basic, just make sure that you know the definitions. These definitions: reflex arc and reflex action. Okay, functioning of a simple reflex action, and then this is one of the examples. So please just make sure that when you revise this, use any other example that you can think of to explain this. All right, and then another thing which is very important, it is the synapse. Please remember. So what is a synapse? It can be defined as a functional junction between the parts of two different neurons. So please remember this. And then the significance of a synapse. So again, please just make sure that you know this. It ensures that the impulse moves in one direction only, and then it also prevents continuous stimulation of the neurons. So that is the synapse. So we have question 1.5. Okay, and then let us look at this question. The diagram below shows a reflex arc. So we have label number A, B, C, D, E, and F. 1.5.1 is like this. Give only the letter of the part that represents the number A, the effector. When you look at this diagram, and then the part that represents the effector is part number E. And then B, the interneuron or connector neuron. So interneuron or connector neuron, it is represented by part number A. And then number C, the sensory neuron. Sensory neuron, remember that it is represented by part number C. So like as I have said, that please make sure that you are able to identify these parts. And then 1.5.2, give the letter and name of the neuron in the diagram that is probably damaged if a person is able to detect the stimulus but cannot respond. So that is letter number F. And then you still remember that letter number F, it represents the motor neuron. And then 1.5.3, state if the nerve impulse travels from D to E or from E to D. And then let us look or go back again to the diagram. They say you have to state if the nerve impulse travels from D to E. Let us look, here is D, and then it means that it will travel like this. It will be D, C, B, A, F, and E. Or does the impulse travel from E to D, meaning that E, F, A, B, C, and D? So please remember that the correct answer here, it will be that the impulse will travel from D to E. And then still under human nervous system. So now we are moving to the parts of the human eye. So please, again, please remember that for you to collect the marks. And then again, you also have to know the diagram of the eye. And then all those parts which are stipulated on your examination guideline and together with the functions of those parts. And then another thing which is very important with the human eye. So please remember accommodation, functioning of the eye, accommodation and pupillary mechanism. And then this is what we have here. The stimulus under accommodation, change in the distance of object from the eye. And then for pupillary mechanism, we have the change in light intensity. And then parts which are involved. What are those parts which are involved in accommodation? So we have the lens, suspensory ligaments, and ciliary muscles. So please remember here, we have the ciliary muscles, not the saccular muscles. So the saccular muscles, we know that they are involved with what? With pupillary mechanism. So with pupillary mechanism, we have the pupil, radial muscles of the iris, and the saccular muscles of the iris. So the main change that must take place, or what is the main change that will have to take place? And then with accommodation, it is the shape of the lens. This is the main change that will take place. And then with the pupillary mechanism, it is the diameter of the pupil. And then what brings about the change above? It is the suspensory ligaments and ciliary muscles. And then on pupillary mechanism, it is radial muscles of the iris and saccular muscles of the iris. So please make sure that, again, you revise these things. Accommodation, you know that we have accommodation for near objects and for far objects together with pupillary mechanism. Please just make sure that you drill, and you drill these things, especially accommodation. So we have accommodation for near visions here. Ciliary muscles contract for near objects, and then suspensory ligaments slacken. So look at the ciliary muscles. Here are the ciliary muscles, and then contracting, and then look at how the suspensory ligaments look. And then after that, the tension on the lens decreases, and then the lens becomes more convex. Look at this part. So it means that the lens is becoming more convex, but with accommodation for distant objects, we will say that the lens, it becomes less convex. I just hope that you see the difference between the two lenses. And then light rays are refracted or bent more, and then after that a clear image is focused on the retina. So this is accommodation for near object. So please make sure that you drill this process together with accommodation for far objects. Okay, we have a diagram here, or a question which is based on this. It is question 1.5.1. Okay, we have all those labels, label number A up to, we also have E, F, G, and H, okay, up to H. And then 1.5.1, it says identify part number A, B, and C. What is part number A? And then when you look at this diagram, and then part number A, it represents the sclera, and then part number B, it is this part, is the cornea, and then part number C, it is the pupil. So please, again, revise the structure of the eye together with all those parts. And then another thing which is very important, you have to know the functions of all these parts. And then 1.5.2, give the letter and the name of the part that number A regulates the amount of light entering the eye. So that will be part number D. This is part number D, which regulates the amount of light entering the eye. And then the name of that part, it will be the iris. And then the second question contains the letter and the name of the part that contains a dark pigment that absorbs excess light in the eye. So that will be part number G. Here is part number G. And then you still remember that part number G, it represents the choroid. And then number C contains the letter and the name of the part that contains receptors sensitive to light. And then that will be part number E, and then which represents the retina. So this simply means that learners, please revise the parts, these parts of the human eye together with their functions. And then we move to the structure of the human ear, still under human nervous system. So another thing which is very important, just make sure that you know this diagram, especially all those parts which are on your examination guideline together with the functions of those parts. And then under the functioning of the human ear, we have a hearing and balance. And then again, please make sure that you drill these two functions, hearing and balance, because definitely one of them, or maybe both, they will be there. They are always there on your examination guideline, on your examination papers. So hearing, you still remember that we have the organ of Corti here. And then with balance, we have maculae and cristae. So let us try to remind ourselves of all these parts that are found on the human ear. So we have question 1.4.1. Identify parts number A and D. Here is part number A. It is the pinna. And then part number D. Here is part number D, semicircular canal. 1.4.2, write down the letter only of the part which, number A, conducts sound waves towards the middle ear. So that will be part number B. Part number B, it conducts the sound waves towards the middle ear. And then number B absorbs pressure from the inner ear. And then the part that absorbs pressure from the inner ear, so you still remember that is part number G, which is the round window. And then ensures equal pressure on either side of the tympanic membrane. And then that is part number F, which is Eustachian tube. And then number D transmits vibrations to the inner ear. And then that will be part number C. Part number C, which are ossicles. And then number E contains the organ of Corti, which is responsible for hearing. That is the receptor which is involved in hearing. And then that is part number E. All right. And then, like as I have said, that please just make sure that you go and revise the process of hearing. Please just make sure that you drill this process. We still remember that I will go through this. The pinna traps the sound waves and directs them into the auditory canal. And then after that, the tympanic membrane vibrates and transmits the vibrations to the ossicles in the middle ear. And then the ossicles amplify the vibrations and carry them via the middle ear to the membrane of the oval window. And then after that, the oval window will vibrate and causes the pressure waves in the inner ear. And then the cochlear vibrations cause the sensory cells in the organ of Corti to be stimulated in the cochlea, and then the nerve impulse are generated. And then after that, the auditory nerve transmits the nerve impulses to the cerebrum to be interpreted. So just make sure that you know this process. And then again, we also have another function of the ear, which is balance. And then this is very, very important. So please just make sure that you don't go on Friday to your trial examination paper 1 without having revised this. And then let us look at a summary of balance. the cristae, which is the receptor in the semicircular canals, are stimulated by the changes in the direction and speed of movement, and then after that the maculae in the circulars and utriculars. Macula, it is also a receptor which is found in the circulars and utriculars, and then they are stimulated by changes in the position of the head, and then when stimulated, and then the cristae and maculae, they convert the stimulus or the stimuli received into a nerve impulse, and then again after that, which are transported along the auditory nerve to the cerebellum to be interpreted, and then the cerebellum then sends impulses to the muscles to restore balance. So please, please, please just make sure that you drill this process, which is balance, or the summary of balance. And then we have a flow diagram here, and then which represents balance. And then, like as I have said, that on this side we have the cristae or crista, and then which is responsible for speed and the direction. And then remember that the cristae, it is found where in the ampulla or of semicircular canals. And then for position of the head, we have the maculae, which is found in the circulars and utriculars. And then after that, an impulse will be generated, and then which will be, or the impulse, the stimuli will be converted into an impulse, okay? And then it will be transmitted along the auditory nerve and then to the cerebellum for interpretation or to restore what? Balance. So again, please make sure that you revise or you drill this balance. Okay, and then we once had a question, and then which was phrased like this. Describe the role of the maculae in maintaining balance. The role of maculae in maintaining balance when a person tilts his or her head to one side without falling over. So this is how you are expected to answer this question. When the position of the head changes, the maculae, you remember that we have said that the maculae, it has to do with what? With the position of the change in the position of the head. So when position of the head changes, the maculae are stimulated. The maculae are stimulated, or you can also say that the maculae, which are found inside the circulars and utriculars, are stimulated, and then after that the stimulus will be converted into an impulse, and then which will be then transmitted to the auditory nerve and also to the cerebellum, and then where impulse is interpreted, and then the cerebellum send impulse to the muscles, and then after that balance will be restored. All right, so now we move to another topic, and then which is response to the environment. Okay, let me do this. All right, response to the environment again, and then here we are having endocrine system. Endocrine system, just make sure that please you know your hormones together with the functions of those hormones. Or you should start first with the glands, and then after that the hormones which are secreted by those glands and the functions of each and every hormone. So we have the ADH here, which is secreted by the hypothalamus, and then after that under the hypothesis, and then we have the FSH. We know that hypothesis, it secretes FSH, TSH, thyroid stimulating hormone, follicle stimulating hormone, and then LH, luteinizing hormone, and then the growth hormone, which is called GH, and prolactin. And then we also have this gland here, which is the thyroid gland, and then it secretes thyroxine. And then adrenal glands, which secretes adrenaline and aldosterone. And then we also have the pancreas, which secretes insulin and glucagon. And then we have the ovaries here. You remember during human reproduction, and then they secrete estrogen and progesterone. And then we also have the testes, which secretes testosterone. So please just remember that you have to be able, or you have to know your glands, and then hormones which are secreted by those glands, and then again the functions of each and every hormone. And then another thing which is very important, please remember to go through or to revise and also drill the following negative feedback mechanisms. And then you remember that I also mentioned one of the negative feedback mechanisms, which is very important, which is that one between FSH and progesterone. So again, please make sure that you also go through or revise or drill the following negative feedback mechanisms: thyroxine levels, blood glucose levels, which is very important, blood carbon dioxide, and water balance, which is osmoregulation. And then at the end, we have salt. So please, 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 learners, just make sure that you revise these negative feedback mechanisms. Thyroxine levels, blood glucose levels. You did this one in grade 11, blood carbon dioxide, water balance, and salt. And then we have the diagram which shows negative feedback mechanism, this one of blood glucose level here. Okay, and then this is what we have again. So under homeostasis, and then you still remember that endocrine system together with homeostasis, and then they will be out of 34 marks. So again, under homeostasis, just make sure that you revise the structure of the skin, and then by using a diagram, and then with an emphasis on the parts which are involved in thermoregulation. And then after that, the role of the following in negative feedback mechanism for controlling temperature or thermoregulation: the role of sweating, vasodilation, and also vasoconstriction. Okay, here is the structure of the skin. Okay, and then we have one of the questions here, and then this question, it is from one of our previous question papers. And then let us try to go through this question. Okay, the diagrams below represent parts of the human skin. Okay, when you look at these diagrams, we have diagram number 1, number 2, number 3. And then look at diagram number 1, okay, blood flow. And then we also have label number A here. And then blood flow again on diagram number 3. So question 1.4.1, it says identify the part labeled A. What is the name of part number A? So part number A, it represents the sweat gland. That will be the sweat gland. And then question 1.4.2, give the numbers of the diagrams 1, 2, 3, and 3 that represent the body's response to high environmental temperatures. And then while we are still on this question, so please again make sure that you revise temperature regulation on a hot and cold day. Please just make sure that you revise that. You can start with hot day, and then you trill, and then after that you know that for cold day it will be the opposite of hot day. So please just make sure that you drill that temperature regulation on a hot day and cold day. So let us go back to our question. Give the numbers of the diagrams 1, 2, and 3 that represent the body's response to high environmental temperature. And then that will be diagram number 2 and diagram number 3. Diagram number 2, the body's response to high environmental temperatures. And then you see here, we have the sweat droplets. And then again, the blood vessels have dilated. Question 1.4.3. Will the skin release more heat through radiation in diagram number 1 or diagram number 3? 1.4.3, and then the correct answer there, it will be on diagram number 3. Give the number of diagram 1, 2, or 3 that represent vasoconstriction. So it means that where the blood vessels have constricted. So question 1.4.4, it will be diagram number 1. The blood vessels have constricted, but when you look at diagram number 3, they have dilated. So the answer for question 1.4.4, it will be diagram number 1, which represents vasoconstriction. So question 1.4.5, which part of the brain controls thermoregulation in humans? And then you still remember that it will be hypothalamus. So please just make sure again that with here you revise temperature regulation, please. In a hot day, you drill, and then after that, you will know that temperature regulation during a cold day, and then it will be the opposite of that one, of the whole day, hot day, please. All right, and then the last topic, plants' response to environment, but now we will be looking at the plants or the plant hormones. So please just make sure that you go through the functions of these plant hormones, please. And then after that, let us move to the role of auxins in geotropism and phototropism. And then again, please make sure that you revise these two. Role of auxin in geotropism, and then again in phototropism. All right, and now we have these questions. All right, let us look at our diagram. We have coleoptile 1 and coleoptile number 2. And then on this diagram here, we have the stem, and then with site number 1 and number 2. And then this clearly means that this stem, it is exposed to light in all the directions. We have light, light, light. And then, but when you look at diagram number 2 or coleoptile number 2, and then we have site number 3 and number 4. And then light from one side. So it means that light shines on site number 4. And then you know what happens here. And then we know that auxin, because auxin is sensitive to light, then it will move to the darker side, which is site number 3. And then after that, the plant or the stem, it will bend towards the direction of the sunlight. And then let us go back to the questions. We have question 1.1.6, which is like this. Which growth hormone is most abundant in the tips of these coleoptiles or shoots? And then the correct answer there, it will be auxin. That will be auxin. And then 1.1.7, after light exposure from one side for several hours, where will the highest percentage of growth hormone referred to in question 1.1.6 most likely to occur? So it will be at number C, number C, which will be 3. All right. And then we have question 2.3. We have an investigation here. Okay. A learner conducted an investigation to determine the effect of auxins and the effect of gravity on root growth in a pea seedling. He used the following procedure, and then on bullet number 1, this is what we have. He germinated the pea seeds for 7 days, and then after that, he then took a sample of 15 seedlings and divided them into 3 groups, A to C, and then of 5 seedlings each. So remember that we have 15 seedlings or seeds, and then which will be divided into groups of 3, which is group number A, B, C. And then each and every group will have 5 seeds. And then on the next bullet, in each group, the 5 seeds were placed horizontally on 3 different clinostats. And then this is the diagram of the clinostat. A clinostat, it is a device which has a disc that rotates at a constant speed. And then a diagram of a clinostat is shown below. Like as I have said that this is the diagram of the clinostat, and then it is rotating, this one which is rotating, and we have the seeds or the seedlings. So, and then after that we continue. He removed the root tips of all the 5 seedlings at the same length in group B. So here is group B. So remember that we have 15 seeds or seedlings, and then in each and every group, and then which have been divided into 3 groups, which is group A, B, C, and then we have 5 groups in each, 5 seeds in each and every group. So group B, he removed the root tips of all those 5 seedlings. No root tips like as it has been indicated on this table. And then in groups A and B, here is group A and B, the clinostats were left stationary. So it means that there was no rotation. And then let us quickly look at our table. So here is group number A and group number B. So the clinostats were stationary. So it means that there was no rotation, and then they were not moving. And then in group number C, the clinostats was allowed to rotate. Here is group number C with a rotating clinostat. But please remember that group number C, the roots are still present, and then no root tips in group number B, and then root tips are present in group number A. And then all 3 clinostats were placed in a dark cupboard. So a summary of the learner's procedure is shown in the table below. And then again on our table we have, remember that we have 15 seedlings, and then which have been divided in group of 3, which is group number A, number B, number C. And then in each and every group we have 5 seedlings. And then group number A, root tips are present, but then the clinostat is stationary. It is not moving or doesn't rotate. And then group number B, again 5 seedlings, and then no root tips, and then the clinostat, again, it is stationary, not rotating. And then on the last group, 5 again, root tips present, and then but now the clinostat is rotating in group number C. After 2 days, the direction of the root growth was observed. And then let us go straight to the questions. We have question 2.3, 2.3.1 up to 2.3.5. Okay, which 2 groups were used to obtain information about the effect of auxins on root growth and the effect of gravity on root growth. Let us go back to our table, and then I will just read our question here while we look that on this table here. Which two groups were used to obtain information about the effect of auxins on root growth? The effect of auxins on root growth, and then that will be group number A and group number B. Group number A with the root tips, and then the root tips have been removed in group number B. So group number A and number B, they were used to obtain information about the effect of auxin. And then auxin was present here, and then no auxin here because the root tips have been removed. So that will be group number A and number B. And then while the clinostat were left stationary. And then question number B, again, which two groups were used to obtain information about the effect of gravity on root growth? And then that will be group number A. Look at group number A and group number C. Why group number A and number C? The effect of gravity. So look here, we still have the root tips. Root tips are present, but now the difference is that the clinostat in group number A, it is stationary, and then in group number C, it is rotating. So it means that group number A and number B, and then they were used to obtain information about the effect of gravity on root growth. And then the next question, explain why the apparatus was placed in a dark cupboard. Explain why the apparatus was placed in a dark cupboard. And then let me show you how to answer this question 2.3.2. Why the apparatus was placed in a dark cupboard. And then it is to ensure that the results are attributed to gravity and not light, or to eliminate the effect of light. So it means that the apparatus was placed in a dark cupboard in order to make sure that the results are attributed to gravity only and not to light. So light has been eliminated. So that is why the apparatus was placed in a dark cupboard. And then question 2.3.3. Describe the expected results for each of the groups B and C in this investigation. And then let us go back to group B and C and look at the expected results. So this is group B and C. Group B and C, no root tips here, and then root tips here. Stationary clinostat and rotating clinostat. And then what will be the expected results? And then let us look at B. No growth will be observed because we know root tips, and then, okay, the clinostat is stationary, but we don't have the root tips. So there will be no growth. And then with group C, roots will grow horizontally, not change the direction. Why? Remember that we have said that with group C, we have the root tips, but now the roots, they won't change the direction because of what? Of the rotating clinostat. So the answer for that, you will just say that the roots will grow horizontally, not change the direction. Because of what? Because the clinostat was rotating. And then question 2.3.4, explain the expected results in group A. And then let us go back to group A. Okay, here is group A with roots present and the stationary clinostat. And then what will be the results of this? let me show you how to answer this question. Question 2.3.4. Remember, root tips are present and the clinostat again, it is also not moving or stationary. So this will be the results. Auxins will move to the lower side of the root. You remember that if the stem is placed horizontally, and then what will happen, auxin will move to the lower side of the root, or if the roots, and then attracted by gravity. And then a high concentration will inhibit growth on the lower side of the roots, while growth will occur faster on the upper side of the root, and then causing the root to bend downwards. And then question 2.3.5. State 3 ways in which the learner ensures a high level of validity for this investigation. So used the same type of plant, and then you have to mention all the things that were kept constant here. The same type of plant was used, and then also again the seedlings were of the same age, and then all groups were exposed to the same environment. And then which was, you remember that they were all placed in a dark cupboard. And then the same number of seedlings for each group. You still remember that we had 15 seedlings, and then after that, each and every group, the 3 groups, and then they had 5 seedlings for each. And then the root tips were cut at the same length, and then all seedlings placed in the same position or horizontally, and then we allowed also the same amount of time for the 3 groups. Okay, and then appropriate controls were set up. So you were only expected to mention the 3. So these are all the factors that were kept constant. All right, so we are on our last slide. This is what we have. Okay, let's take hands and learn these key points so that we can pass and we can all smile. And then that brings us to the end of our presentation. But before I leave, let me quickly make a summary of these key points, please. So I will just jump this format of the paper. So please remember that all these key points, just make sure that you go through them or you drill them, and then definitely they will help you to pass, especially those learners who fall under level 1 to 3. And then remember that I have mentioned the issue of reproduction in vertebrates, like as I have said that it will be there on your next coming trial examination, and then also on your final examination, and then still the mark allocation will still be 8 marks. So please just go and revise this. External, internal fertilization, I know that we don't have a problem with these ones, and then amniotic egg, and then please again remember precocial and altricial development, and then parental care. And then please, these 3 again, go through these 3 and then make sure that you know this: ovipary, ovovivipary, and vivipary. And then again, we also talked about, okay, under human reproduction. So please remember, we have mentioned that you have to know the structure of the male and female reproductive system, together with all the parts which are stipulated on your examination guideline and the functions of those parts. And then another thing which is very important on the male reproductive system, Make sure that you know all these parts together with their functions, not forgetting your glands. Please, you have to be able to identify the location of your glands together with the functions of these glands. And then again, location of the epididymis and the function of it. And then again, please remember the issue of the testes and the scrotum. So please, you have to be able to explain that. And then again on them, okay, the structure of the sperm cell and the structure of the ovum, they are also important. And then again, the process of spermatogenesis. Please, the process of spermatogenesis and oogenesis. And then, like as I have said, that you are not allowed to describe this using your own ways. So it simply means that you will have to sit down and then revise this or drill these processes. So you will see these things when you write your examinations. And oogenesis, it is always there, especially during the final examination. But please just make sure that you also prepare this for trial examination. And then again, the hormonal control of the menstrual cycle. And then you remember, I also mentioned that you have to know your 4 hormones: FSH, estrogen, LH, and progesterone. Please, please, please. And then again, the negative feedback mechanism involving FSH and progesterone. This one for sure, you will see it on Friday on your trial examination. And then again, on your final examination, it will still be there. It is always there on your examinations. So the negative feedback mechanism involving FSH and progesterone. So just make sure that you go and revise this or you drill that. All right. And then again, another thing which is very important, development of a zygote. Remember the development of a zygote up until it reaches implantation. Please remember this, and then also this for final examination, but also prepare this for the trial examination. And then you still remember what I have said here. You remember fertilization takes place, and then after that we have the zygote. And then the zygote, which will divide by the process of mitosis, and then it will divide up until this stage, which is called morula. And then also, like as I have said, that please remember that morula, it is a ball of cells, not a hollow ball of cells, but you only say hollow ball of cells when you talk about the blastula, because of this hollow space which is found inside. So this is how you write that in an exam. This is very important. Please just make sure that you drill this. After fertilization, remember that the zygote will undergo mitosis or divide by the process of mitosis, and then to form a ball of cells called morula. And then morula will divide again by the process of mitosis to form a hollow ball of cells called blastula, and then which will form the outer layer called the chorion. And then chorion, you know that it will form what is called a chorionic villi, and then after that implantation will take place. So please revise this or drill this process. And then again, another thing which is very important for the next coming examination, know the functions of this chorion, chorionic villi, amnion, amniotic fluid, and then the placenta, please. And then also the umbilical cord, and then which contains the veins and arteries, please again. And then the placenta, the role of placenta during the process of pregnancy. So please just make sure that you go through this. All right, and then under response to the environment, human nervous system, remember location and functions of the following parts of the brain. The cerebellum, corpus callosum. Please, please, please just make sure that you revise this. You have to be able to identify those parts on the diagram, and then again be able to mention the functions of those parts. Cerebrum, corpus callosum, medulla oblongata, please, and then again the spinal cord. And then another thing which is very important, the neurons, motor neuron and sensory neurons. So please revise those structures. And then we move to the next. Okay, reflex action by using any other example. So please just you have to sit down after this, and then you drill this. And then you remember again, I have said that you have to be able to identify these parts. We have the receptor, sensory neuron, interneuron, motor neuron, and effector. Please. And then another thing which is very important, where you can collect marks, make sure that you know the definitions, these definitions of the reflex arc and reflex action. Okay. Function of a reflex action using an example there. And then the synapse, please make sure again that you know this, which is a functional junction between the parts of two different neurons. And then again, please, the significance of the synapse. Okay, and then another thing which is important under the human eye, the structure, those parts together with their functions, please. And then another thing that you have to know, the functions of the eye. You remember accommodation for distance and near objects and pupillary mechanism. So please just make sure that you drill this accommodation and pupillary mechanism. Just drill this, please. All right, and then under the human ear, again, the structure, the parts, and the functions. And then after that, functioning of the human ear, which are hearing and balance. Please go through these two functions. Okay, and then this is a summary of hearing. I know that you don't have a problem with this one. And then balance is a problem. So please just make sure that you drill this. This is the summary of balance, please. And then we have a flow diagram here. But please remember that maybe if a question is phrased like this or appears like this, describe the role of ear in balance, then please, you are not allowed to use the flow diagrams. And then you just have to describe it in this way. And then again, we have the glands under response to the environment. Again, you still remember that we have endocrine system and homeostasis. So under endocrine system, remember your glands. Remember the glands, the hormones which are secreted by those glands, and the functions of those hormones, please. And then another thing which is important here. Here is another thing which is important. Please make no mistake of going to your examination, the trial examination that you will write on Friday, without having revised this. The negative feedback mechanisms, they are very important. We have the first one, thyroxine levels, and then blood glucose level. And then this one, you did it in grade 11. So please just make sure that you go back or you sit down and then you drill this one, blood glucose level. And then blood carbon dioxide, water balance, you also did this one in osmoregulation. Revise, drill. And then again, we have this one of salt. So please remember that negative feedback mechanisms, we have the first one, which is between progesterone and FSH, and then you remember that I have said that you have to know that one for both the trial examination and the final examination. And then again, we also have these 5 negative feedback mechanisms: thyroxine levels, which is new in grade 12, and then blood glucose levels. Drill this one because we also did it in grade 11. Blood carbon dioxide, you also did this one. Water balance, drill, revise, and then the salt. And then under thermoregulation, the structure of the skin, you remember what I have said here. And then another thing which is very important with this one, remember temperature regulation on a hot and cold day. So please make sure that you revise that and you drill that. You can decide to start with this one of hot day. You will drill, and then after that, you will just have to know that cold day, and then it is the opposite of hot day. So please just make sure that you revise that. Okay. And then plant hormones, the functions of the hormones. Revise the functions of these 3 hormones, and then the role of auxin in geotropism and phototropism. Please, again, you have to revise that. All right. And then again, this is the last slide. Let's take hands and learn these key points. You still remember that all these key points, these are the basic things that will help you to pass this coming examination. Please. All right, and then that brings us to the end of our presentation for paper 1. Paper 1, preparation of trial examination. All right, thank you so much.